Good morning. Today I'm going to talk about the development of the respiratory system. But before we start talk about the development of the respiratory system, I will talk a little bit about the anatomy. If you remember, we talked about the anatomy of the respiratory system. We said it divided into two portions. We have the conducting portion and we have the actual site where the gas exchange occurs, which is the respiratory portion. As I said, the conducting portion start from the area of the nose, external nares, then we have the nasal cavity, then we have the area of the pharynx, then we have the area of the larynx and the trachea, then it divided into 10 main bronchi, and these bronchi further divided inside the lung until we reach to the level of the terminal bronchioles. All of these structures are considered as a conducting portion, whereas the area where we usually have the gas exchange, which include the respiratory bronchioles, alveolar duct, alveolar sac, and alveoli, all these structures are considered as the respiratory portion. So again, this is the area, all of this area here, where just only conducting the air toward the lung is considered as a conducting portion, where as the area here starts from the respiratory bronchioles to the alveolar ducts, then we have the alveolar sac, then we reach to the level of each alveolus. All of these are the respiratory portion. So I'll talk a little bit about the formation of the nasal cavities. Okay, at the beginning we need to know that the development of the face include formation and battering of the 5 facial primordia, and this is responsible for the formation of the orbit and the nasal and the oral cavity. We have the first one here, which is the prominence, which called the frontonasal sinus, and this usually develop in the area of the telencephalon. Then we have here we have the maxillary process or processes or maxillary prominence, okay, and the ventral one here, which is called the mandibular processes or prominence. As we know, these mandibular processes fuse together to form the lower jaw, and usually these maxillary processes are fused together to form the upper jaw, which include the maxillary and the incisive bone. And at this area here, we have the area of the frontal sinus, which would be responsible to the formation of the nasal cavity and also formation of the area of the orbit. The structure that we can see here, which is the stomatium, okay, this is structure, this is a membrane, usually close the opening of the oral cavity. So this area here usually will be closed by a membrane, stomatium, later will atrophy, and this will result in the formation of the oral cavity. So from this we can see that this maxillary structure with the frontal structure here will form the area of the upper jaw, where the one that found here below, which is the mandibular processes, will help in the formation of the lower jaw. At the area of the frontal nasal prominence, which is, as I said, developed in the area of the telencephalon, which is the area of the forebrain, there will be two ectodermal thickening. One of them will be the nasal prominence, okay, and this will result in the formation of the nasal cavity, and the other one, which is the lens placodes. So we have the nasal placodes and we have the lens placode, which is an ectodermal thickening. As I said, the nasal placode will result in the formation of the nasal cavity, whereas the lens will result in the formation of the lens of the future eye. So here, this is the area of the frontal nasal prominence, this is the maxillary, this is the mandibular. In the area in the frontonasal, this is the area of the nasal placard. As I said, it's ectodermal thickening. At this area here, there will be proliferation of the underlying mesenchymal cells. The mesenchymal cells that is found just below the ectoderm start to proliferate, and their proliferation will result in the formation of this structure here, okay, which is almost like the horseshoe, okay, and this horseshoe have a lateral edge, which is the lateral nasal prominence, and have a medial prominence which is called the medial nasal prominence. And at the area here in the middle between the lateral and the medial, this is the area where we'll have the nasal pits, which is the area that will going to be deepen and result in the formation of the nasal cavity. So we can see at this area here, this is the area of the right and the left nasal cavity. They start as a separate, and we can see they come close together to at the end form the area of the nose area, and we have in the area here we have the opening of the nasal cavity, which is the external nares, and we can see this structure here, which is the future eye. At the early stage of the development, we can see here there is a groove between the maxillary and the nasal prominence, and this groove is called the nasolacrimal groove. And we can see here this nasolacrimal groove extend to the canthus, nasal canthus of the developing eye. And usually at this area here there will be thickening of the ectoderm, okay, and there will be a formation of the solid cord of cells which invaginate deeply into the underlying mesenchymal tissue. And at the end, this ectodermal cord will lose the connection with the ectoderm. It will be found just in the area of the mesenchyme. And this cord will undergo programmed death and a process called canalization, which is the formation of the canal inside this cord. And this will result in the formation of the nasolacrimal duct. If you remember, the nasolacrimal duct extend from the eye to the nose. So the origin of this one was ectodermal cells. These ectodermal cells form a cord between the maxillary and the nasal prominence. Then these cells will lose their connection and develop in the underlying mesenchymal tissue. Later there will be a process called canalization, which is a canal inside this cord through programmed death, which is apoptosis, and this will result in the formation of the nasolacrimal duct. So as I said, this is the area of the horseshoe. We have the lateral and we have the medial prominence of the nasal area, okay, and we have here at the middle we have the nasal pits, okay, and we can see this horseshoe structure result due to the proliferation of the mesenchymal tissue, okay. So these nasal pits, which is surrounded by the medial and the lateral prominence, gradually invaginate the underlying mesoderm of the frontonasal sinus. And this is the area of the nasal placard at early stages. As I said, this start to deepen, so it start to go inside the area of the frontonasal prominence and start to enter the area of the mesenchyme. This nasal pits, as I said, it start to deepen and this result in the formation of the nasal sac, okay, and there will be right and left nasal sac, and usually they are separated from the oral cavity through a membrane which is called the oronasal membrane. And this oronasal membrane form the primary palate. We know that between the nose and the oral cavity there is a separation, and this separation is by the hard palate. So early this membrane, as I see here, this called is the oronasal membrane, which is the primary palate. And you can see here, this is the nasal placard, and start to deepen and elongated caudally, and this result in the formation of the nasal sac. As I said, there is a separation between the nose and the oral cavity through this membrane, which is the oronasal membrane. As I said, it will play a role in the formation of the hard palate. Later at these stages, the caudal portion, which is the partition between the right and left medial wall of the primitive nasal sac, will atrophy, and this will result in the formation of the common nasal cavity. Okay, so if we go here, you see at this area here, just go back here. At the beginning, we have two separate area for the right and the left nasal cavity. So the caudal portion will atrophy, and this result in the formation of the common nasal cavity. And the second important thing is that the caudal portion of the primary palate will atrophy. So this area here again will undergo the atrophy. So this here, we can see that this is the area of the nasal cavity, and we can see here that the right and the left nasal cavity will communicate with each other through the atrophy of the caudal portion of the nasal septum. And the second important thing, that the caudal part of the primary palate will atrophy, and this will result in the formation of the common area for the respiratory portion between the right and the left nasal cavity, and this structure is called choana. So at this area here, we can see here we have the area of the secondary palate, and later we'll talk about how it's going to be formed. So if we go back, we have the area of the nasal placodes. This going to deepen. This will result in the formation of the nasal cavity. This nasal cavity or nasal sac will be separated from the oral cavity through this membrane, which is the oronasal membrane, which result in the formation of the primary palate. And we can see here, this is the area of the digestive system, and this is the area of the developing tongue. So this is the area, we can see this is the area of the nasal cavity, and this is the area of the oral cavity. We can see here that the tongue is projected in the cavity of the nasal cavity. So the tongue is found in the oral cavity and in the area of the nasal cavity. With the development of the tongue will no longer be projected in the nasal cavity. We can see here this structure here, which is the lateral palatine process. We have the right and the left. So when the tongue is no more projected in the nasal cavity, this allowed the expansion of these lateral palatine processes, and this will result in the establishing two cavities. We have the nasal cavity and we have the oral cavity, and we can see that these two lateral palatine processes, they are come together and fuse together. Okay, and this fusion will result in the formation of the secondary hard palate. So the primary palate is formed by a membrane, which is the oronasal nasal membrane, whereas the secondary is formed due to the expansion of the lateral palatine processes, the right and the left. As I said, they fuse together. And the second important thing we can see here, this is the area of the nasal septum, which, as I said, is divided the right and left nasal cavity. This one will grow ventrally, ultimately will fused with the hard palate, and this will result in the formation of the nasal septum and also in the formation of the hard palate, which separate the nasal cavity from the oral cavity. So as I said, the nasal septum will fuse with the area of the lateral palatine processes. This will result in formation of the right and the left nasal cavity, and also result in the formation of the nasal cavity, which will be separated from the oral cavity. Okay, very important thing we said that about the caudal portion. Usually the nasal septum is atrophied, so we have a common nasal cavity for both right and left nasal cavity, and as I said, this is, we call it the caudal nares, or in a human they call it posterior nares, but in the animal we call it caudal nares or the cavani. However, in the horse, this is very important thing, there is no caudal atrophy of the nasal septum. So each of these nasal cavity will communicate with the area of the pharynx by a separated opening. This is very important. There is no common area of the nasal cavity in the posterior portion of the nasal cavity in the horse, whereas in the other animal, we have this communication. So again here, just to remind you, we have the frontal nasal prominence, we have the maxillary, and we have the mandibular. As I said, mandibular will result in the formation of the lower mandible. The maxillary will result in the formation in the upper jaw, which include the maxillary and the incisive bone. And as I said here, this is the frontal nasal prominence, which have two thickening, which is the lens lacude. Also we have the nasal pits. Here this is the area of the horseshoe area. We have the lateral margin and we have the medial margin, and as I said, this area is formed due to the proliferation of the mesenchymal tissue just below the level of the ectoderm of the nasal area. This is the area of the nasal pits. This pits, as I said, will deepen and elongated caudally to form the nasal cavity. We can see here, this is the area of the nares. See, they develop separated, then they come close together, ultimately they fuse together to form the nose area. Okay, we'll talk about the development of the concha. As we know, there's a dorsal concha and we have the ventral concha, and in the caudal portion in the animal we have the middle concha. And these concha, usually they have longitudinal lamina which arise from the nasal bone and from the maxillary bone. Usually these concha a shelf-like projection, okay, and later this shell take the scroll-like conformation, okay, and usually from outside this lamina is surrounded by ectoderm, and usually this ectoderm later will differentiate into the respiratory epithelium, which is a typical pseudo-stratified columnar epithelium, whereas the core, which is in the middle of this lamina, we have mesoderm, and these mesoderm at the beginning they will differentiate into cartilage through formation of the primary quantification center, and later these cartilages will undergo the process of the endochondral ossification, and this cartilage will replaced by a bone. For the paranasal sinuses, as we know, we have two important sinuses. We have the frontal sinuses and we have the nasal maxillary sinus. As we said in the anatomy, it's just a diverticule of the nasal cavity into the bones, which is mean as invagination of this bone and formation of this cavity, and this cavity they have a common opening with the nasal cavity. Okay, usually these nasal sinuses, which is the paranasal sinuses, is developed by epithelial growth. The epithelium in the area of the nasal cavity will grow inside the bones. We know that the bone of the skull is a flat bone. We have two layers of compact bone. At the middle we have a spongy bone. So this epithelium will grow into the bone, and this will result in the formation of the cavity. And this cavity, as I said, they keep their opening with the nasal cavity. The cavity between the bone is called diploe, which means there's, as I said, there's a spongy layer between inner and outer compact layer. And usually these paranasal sinuses will be filled with air. As I said, we have different paranasal sinuses, but very important to know that these paranasal sinuses keep their contact and communication with the nasal cavity. And the origin of these epithelium that found inside these paranasal sinuses come from the ectoderm of the nasal cavity. Now we are going to talk about the development of the larynx, trachea, bronchi, and the lung development. Early in the development, we know that the foregut is formed before the formation of the respiratory system. Usually, the respiratory system is developed from the ventral portion of the foregut. So the primordia of the respiratory system develop as a ventral groove. See this groove here from the foregut, the floor of the foregut. This groove is called laryngotracheal groove. This groove, as we can see here, going to deepen, then going to be elongated, which form elongated outgrowth, and this extend caudally. Later, there will be a separation between the foregut and the primordia of the future respiratory system, which is the area of the laryngotracheal tube. And this separation is going to happen by these two grooves, one in the right, one in the left, which is called the tracheoesophageal groove. And we can see here this tube keep elongated outgrowth caudally. Okay, at the end, this groove, the right and the left one, going to meet, and this will result in the formation of the structure here, which called the tracheoesophageal septum. So at the beginning, it will be a groove. When this groove, the right and left side, meet together, this will result in the formation of the septum. And usually this septum separate dorsally the foregut from the ventral portion, which is the tracheobronchial tube, which is laryngotracheal tube. We can see here the cranial portion. Here at this area here will result in the formation of the pharynx, whereas the cranial portion of this tube, which is the laryngotracheal tube, will result in the formation of the larynx. So again, this is the foregut, this is the ventral growth, of the respiratory portion, which is called laryngotracheal groove. And you can see this tube grow caudally, then start to separate through the esophageal tracheal groove, and these come together, result in the formation of the tracheoesophageal septum. As I said, the cranial portion from each tube, this one, the foregut, will result in the formation of the pharynx, whereas the other portion will result in the formation of the larynx. This tube continues to grow caudally as elongated outgrowth, and this will result in the formation of the larynx, trachea, and also we can see here that the caudal portion going to be divided into two structure. It's almost like bud off from this tube here, and this will result in the formation of the bronchial buds, which going to form the two lungs. As I said, the larynx develop from the cranial region of the laryngotracheal tube, communicate with the primordia of the pharynx. The epithelium of the larynx is derived from the foregut endoderm, while as the cartilage and the muscle of the larynx forms from the splenic mesoderm. So we have endoderm of the foregut, just responsible formation of the epithelium with the glands, whereas the mesoderm that going to surround this tube later going to develop all the cartilages, all the muscle and the connective tissue that surround the area of the larynx and the future trachea. So this is the area of the larynx. If you remember, we said that the larynx, we have cartilages, which is the thyroid cartilage, we have the epiglottis, we have the cricoid, and we have the arytenoid. And we talk about the muscle that we have in that area. We have the extrinsic muscle and we have the intrinsic muscle. Extrinsic muscle that extend from the larynx to the other organ, whereas the other muscle which connect the different cartilages within the larynx is called the intrinsic muscles. So we can see here, this is the area of the laryngotracheal tube. As I said, we have the epithelium here, which is the origin from the endoderm, whereas the splenic mesoderm going to surround this endoderm later. We can see here that there is a bifurcation of this tube here, which will result in the formation of two bronchial buds. And we can see here that the cranial portion result in the formation of the larynx, and the caudal portion at this area here will result in the formation of the lung, whereas the area that we have here in the middle will result in the formation of the trachea. Now we talk about the tracheal and bronchial development. The endodermal lining of the tubes give rise to the respiratory epithelium, mucosa, and submucosal gland, because if you remember that the mucosa is consist of the epithelium, lamina propria, okay, and the glands, their actual origin from the epithelium, and then they undergo the growth to the sub-underlying connective tissue. We can see here that the area of the buds, which is the bronchial buds, going to enlarge and start to divide, and they grow at this area here. See this membrane that we have here, which is the area of the pleuroperitoneal membrane. And we can see here that the one that found directly in the lung tissue result in the formation of the visceral layer, whereas the layer that found outside will be the parietal layer, and the space between them, that will be the pleural cavity. So we can see here again, this is the bronchial bud start to divide into two branches or three branches, depend if you have the right or the left lung. If you remember when we talk about the lobulation of the lung, for example, in the ruminant or in the dogs, In the right side, we have cranial, we have middle, and we have caudal, and we have the accessory loop. Whereas in the left side, we have just only the cranial and the caudal loop. And if you remember, in the horse, we only have three loops, which is the cranial, the accessory, and we have the caudal, whereas the middle one is absent. We can see here that bronchial, this is going to divide. This is the main bronchi. Then we have the lobar, and this lobar will further divide it and bifurcate. And remember, every time we have bifurcation, there will be two new tubes, and also the diameter of these tubes will reduce. So we can see here, this is the trachea, the mini bronchi, then we have the lobar start to divide, then we have secondary, tertiary, then we start to reach the level of the bronchioles, and we have primary bronchioles, and we have finally the terminal bronchioles, which consider as a part from the conducting portion. The last portion, which is the respiratory bronchiole, this is not found in all animals, it's only found in the primates and carnivores. This one consider as a transition between respiration and conducting portion. One important thing that in ruminant and pigs, the right cranial lobar bronchus give off directly from the trachea, and this referred to as tracheal bronchus. And this is very important to differentiate between ruminant and pigs from the other animal species. Remember, we have the tracheal bronchus, which is a direct branch from the trachea that's going to supply the right cranial lobe. As I said, the lobar bronchi will give off the tertiary. Then we have the segmental bronchi. Just we go to this again. You can see here, this is the main bronchi, start to divide, and this is the area of the segmental bronchi. As I said, the respiratory bronchioles are transitional zone between conducting and the respiratory region, because this portion, the respiratory, there is some gas exchange, also conducting portion. Whereas in the area of the primary and terminal bronchioles, this portion is considered as a terminal bronchioles. Terminal bronchioles are considered as a part from the conducting portion. Now we are going to talk about the development of the lung. For the development of the lung, usually we divide the development of the lung into five stages. Okay, and this depends mostly in the histological feature. We have the embryonic stage, we have the pseudo-glandular stage, we have canicular stage, we have the terminal sac stage, and finally the alveolar stage. So we start with the first one, which is the embryonic stage. This stage extends from the formation of the laryngeotracheal groove, which means from the beginning of the development of the respiratory system until the formation of the segmental bronchi, which we said that the main bronchi are going to divide to form the lobar, primary, secondary, tertiary, and then finally the segmental bronchi. And usually we'll have like between 12 to 18 division to reach to this level of the segmental bronchi. And as I said, the lung going to develop within a common pleuropericardial cavity. At the beginning, the pleural cavity and pericardial cavity, there will be like a common cavity. Later, they are going to divide into pleural cavity and pericardial cavity. And as I said, they will be surrounded by visceral pleura, and we have the parietal pleura. The second stage we have, which is the pseudo-glandular stage, and this extends when the developing lung, which is the bronchial bud, is extended into the surrounding mesenchymal tissue. And because the appearance similar to the pseudoglandular or the glands or exocrine gland, they call it the pseudoglandular stage. At this stage, by the end of this stage or this period, we have 14 segmental bifurcation has occurred. And all these are major conducting branches of the bronchial tree are formed. So from this level, all the primary, the secondary, the tertiary, and the segmental bronchi is developed. Furthermore, the primary and the terminal bronchioles are formed. And we can see this epithelium, the endoderma, is surrounded by this mesenchymal tissue, which will result in the formation of all structures that surround the respiratory organ. For example, we have the connective tissue, we have the muscle, we have the cartilages. And at the beginning, this epithelium, all of it are cuboidal epithelium. Later we'll go on differentiation for respiratory epithelium or Clara cells or goblet cells. The third stage, which is the terminal sac stage, at this stage we start to develop large number of the terminal sacs. And all these terminal sacs are bud off from the respiratory bronchiole. So you can see all the structure here, all of it. See the structure here, and this is the bud off, and this will result in the formation of the terminal sac. Each of these terminal sacs are lined by cuboidal epithelium, and remember from the histology we said that these alveoli surrounded by or covered or lined by the epithelium, which is alveolar type 1, which is simple squamous epithelium, whereas type 2, which consider as about 5% of the total cells that found at that area, are simple cuboidal epithelium. So most of this epithelium will change from the cuboidal epithelium into squamous epithelium. The fourth stage, which is the canicular stage, canicular canal. So at this stage, the lumina of the bronchi, the bronchioles enlarge. So this is an enlargement of the cavities. Okay, as I said, there is also this bronchiole give us the respiratory bronchioles. As I said, there's the formation of the terminal sac. The last stage we have, which is the alveolar. At this stage here, as I said, we have the mesenchymal tissue surround. This mesenchymal will differentiate into the blood vessels, the blood capillaries. And we can see at this area here that this blood supply will be in close intimate and attachment to the alveoli. And this will result in the formation of the blood gas barrier. And this is the last stage of the development of the respiratory system. So if we go back, there are 5 stages. We have the embryonic, we have the pseudo-glandular stage, we have canicular, we have the terminal sac stage, and finally we have the alveolar stage. As I said, embryonic extend from the formation of the laryngotracheal groove until the formation of the segmental bronchi. The pseudo-glandular, we can see here that these formed segmental bronchi start to invade the area of the mesenchyme, so that's why they call it the pseudo-glandular stage. Then we have, before that, we have the canicular stage, where there's like enlargement of the cavity of these, and the fourth stage, which is the terminal sac stage, where we have these develop at this area, which start to bud off from the respiratory system. And the final stage here, as I said, the mesenchymal tissue here start to differentiate, and this will result in the formation of blood vessels, and these blood vessels will be in close contact with these alveoli and result with the formation of the blood-air barrier. Now we'll talk about the postnatal growth of the lung. The respiratory system will not be fully developed within the embryonic development. Some structure or some stages happen after the postnatal development. Usually the postnatal growth of the lung is marked by the formation of additional alveoli or by subdivision of the existing alveoli. And after the division of these alveoli, there will be a formation of the septum. And this all very important to increase the surface area for the gas exchange. Just remember that during fetal development, the lungs are filled with the fluid because the body don't need it. There's no way that he can do the respiration. But after the parturition, the lung will be emptied from this fluid and start to perform the respiration function. Now, just to remind you about the divisions of the lungs. Okay, for example, here we have the canine, we have the right, we have the cranial, we have the middle, the caudal, we have the accessory, and each one of them they will be supplied by a separate lobar bronchi, whereas the left lung will be supplied by two lobar bronchi, and this mark their division, which is the cranial and the caudal. Remember this structure here, which is come directly from the trachea and supply the right cranial. This is found in ruminant and pigs. In the horse, we can see we have the cranial, we have the caudal, we have the accessory, whereas in the left side we have only cranial and caudal. The last thing I want to talk about, which is the septum transversum, which is responsible for the formation of the diaphragm, which separates the thoracic cavity from the abdominal cavity. Usually this structure, which is the septum transversum, develop caudal to the heart, and this results in the formation of the body wall after the lateral body folding is completed. You remember that we have the ectoderm, mesoderm, we have 3 layers. Then we have to do the folding, and this result in the closing of the body cavities from the ventral portion, and we have the cranial folding and the caudal folding that will result in the formation of the tubular structure of our body. As I said, the septum transversum will result in the formation of the diaphragm, which separates the thoracic cavity from the abdominal cavity. This diaphragm develops from 4 structures. We have the lateral body wall mesoderm, we have the septum transversum, we have the dorsal mesentery of the esophagus, and the final one, which is the pleuroperitoneal fold. All of these structures will result in the formation of the diaphragm. This structure here, which is the septum transversum, we have the pleuroperitoneal membrane, we have the body wall mesoderm, and finally we have the esophageal mesentery. All of these 4 structures will help in the formation of the diaphragm.